Should we use animal testing for biomedical research? No. Although animal testing has led to past medical advances, in the future we should use alternative methods to biomedical research. I'm going to be exploring this topic through ethical and scientific lens. First of all, what is animal testing? Animal testing, according to the Humane Society International, is defined as procedures performed on living animals for the purposes of research. This method of experimentation has been used since 500 BC. Subjects are exposed to chemicals, drugs, diseases, genetic mutation, and surgical procedures. A multitude of species all around the world makes up the more than 115 million animals used for biomedical research a year. The objective of these experiments is to improve knowledge of human health and human biology, but the recent studies about the effectiveness of these procedures leads us to believe, is this the best form of research? Data shows that animal testing studies fail to predict real human outcomes in 50 to 99% of cases. This is a quote by Humane Society International. Laboratory environments, inconsistent genetics, and difference in device and diseases modeled by animals are all factors that could lead to different results when translated to human trials. According to the National Library of Medicine, the FDA estimates that 92% of drugs that pass preclinical tests never make it to market, as well as 114 new potential therapies that were initially tested on animals that worked that did not in human trials, resulting to humans leading to cancer, brain injury, and death. Failing to transfer successful biomedical research to humans is ending in massive amounts of animal waste and the ultimately non-beneficial outcomes to humans. Scientific concessions. You must be thinking, is animal research all that bad? No. National Academic Press states that animal research has even contributed to better nutrition and sanitation since it has helped to identify that agents that contribute to good and bad health. Other positives that have stemmed from animal testing are balloon angioplasty, which was developed using dogs. Tests for mania were used testing on guinea pigs. Mice have had a vital role in research of organ transplant and bone marrow transplants, as well as vaccines and antibiotics went through the same testing. Each one of these major medical advances has saved and improved lives of millions, and concessions about these discoveries could be proved impossible without the experimentation on non-human subjects. But do past results make up for the future of this outdated method? Around 126.9 million animals are bred, tested, and killed for cosmetic, chemical, and household product industries a year. Animals participate in forced chemical exposure for toxicity, infectious disease, and surgical procedures, as well as infliction of burns and other injuries to study healing. There are also behavior experiments that purposely cause stress by design, including electric shock and forced swimming and running. In order to perform these procedures, animals have to be prepared, receiving ear notching and tail clipping for identification, food and water deprivation, and short and prolonged restraint, as well as inadequate living conditions for where they have to recover from these experiments. In each situation, these animals go through traumatic experiences, creating more valuable variables to the experiment and further affecting the study. Quote, drug failures and research that never deliver because of irrelevant models that not only delay medical process but also waste resources and risk health and safety of volunteers in the clinical trials. Humane Society International 2012. Ethical concerns about the nature of these procedures lead to continuous questioning of the morality of subjecting these animals to the abuse of testing and the results that humans have to face because of the consequences. If not animals, then what? Other methods of experimentation have become more relevant because of the questioning of animal testing. There is many alternatives, such as testing cells in test tubes or cell cultures, 3D tissue cultures, or otherwise known as organ on a chip, stem cell research, computer and mathematical models, and non-invasive diagnostic imaging. Each one of these alternatives are being used and created, moving biomedical research away from the unreliable results of animal testing. With all the fail rates of animal testing and promising new technology, we should continue to push for more ethical ways to research. Although animal testing has led to medical advances, in the future, we should use alternative methods for biomedical research.
All right, so a couple of questions. What information did you need that you weren't able to find or locate, and how did you go about trying to find that information?
I would just say some of the new information about organ on a chip, because we don't have like testing and like information as we do for animal testing. We have all these statistics about the fail rates and the pass rates of animal testing, but all these new developments, like even computer models and stuff, we just don't have the same research to support whether they work well or not. So we do have some things saying that, oh, they're going to be really prevalent in the future, but we don't have like hard facts like we do for animal testing.
Okay, thank you. What additional questions emerged from your research, and why are they important?
I would say just, I would go for, I would say like the future of the new processes, how we're going to animal test. I think that the new technology sounds really good and beneficial, but the question rises, what are we going to do with all the animals that are still in captivity from these animal testing methods that we've used in the past, and what the transition is going to look like? Because if we just end up killing and burning and hurting our environment more to get rid of old ways, then is it really beneficial to move into new technology super fast?
Okay, thank you.